Up till now, everything that we've been lecturing on, discussing, have been inanimate objects. Whether it's plastics, metals, woods, things like that, things that have a surface. What happens with something that you need to sterilize that's a liquid? Something you need to sterilize that's a gas? Well, you could heat them, which unfortunately for a lot of chemicals can damage the chemicals, to the point that they're no longer have any activity. So whatever compound you had in a solution is now basically destroyed. You heat some gases, you'll get some chemical reactions, and well, that's not good. So what we've developed is a method of sterilization referred to as filtration. You've probably heard the term in advertisement and things like that. Oh, use blah, blah, blah. It has HEPA filters. HEPA filters are filters, whether they're plastic or paper-based, that has microscopic holes, kind of like a sieve. But as you look here in not only the artist drawing, but the electron microscope picture, the holes are too small for the microbes to fit through. Whether the microbes are viruses, whether the microbes are protozoan or bacterial, different filters have different size holes. The more sterilization, levels of sterilization you want, or disinfection to sterilization you require, you get smaller filters with smaller and smaller holes. So the liquid and the solution, whatever's in the solution, the chemicals, the atoms, the molecules, freely pass through, but any cells present, any endospores present, will be caught in the filter. Whether it's liquid, whether this is gas, same thing. Downside to this, though, is when you're talking some medications, especially ones that are going to be injected into a human, into an animal, you can sterilize the medication. You can sterilize the liquid relatively easy, pass it through a filter, no living cells present. The problem is these filters do not stop proteins, do not stop endotoxins. Remember LPS, endotoxin from the bacterial cell. Part of the bacterial cell comes off. Lipopolysaccharide. Lipopolysaccharide can be more damaging to you health-wise than having an active bacterial infection. Next time you're in a doctor's office or a hospital and you see the little vials that they're taking the medication out of to inject, you look at it, you read the label, it'll say endotoxin tested. So the liquid has been filtered, and then it's been tested to make sure that none of those endotoxins, none of that LPS is present. It's filtered just in case something got in there, some fungal, some protozoan, some virus, and then it's endotoxin tested to make sure that no bacteria were present during the making of that medication. Little tip, little tidbit to keep in mind.